Hi, my name is Ali Riazi. I'm co-founder and CEO of Theravac Biologics. Thank you for the opportunity to present to your group. At Theravac, we are developing innovative active immunotherapies for neurodegenerative diseases and Alzheimer's. The leading neurodegenerative condition, obviously, is a huge unmet need. And the number of affected people is projected to reach 150 million by 2050, presenting a multi-billion-dollar market opportunity for drugs that so far has been mainly addressing the symptoms and not disease-modifying. But there is a glimmer of hope for patients like Nancy, who was one of the first people receiving monoclonal antibodies recently approved by FDA. Leqembi and Kisunla is the second one that was recently approved also. But these antibodies come with drawbacks. Patients need to go to a hospital and get infused with hundreds of milligrams of antibodies every 2 or 3 weeks, and stay on these treatments for relatively a long time. These antibodies occasionally come with serious side effects, such as brain inflammation and bleeding, and they're very expensive. And despite all that, the benefit to patients is about only 25% reduction in the rate of the disease progression. So at Theravac, we are aiming to change that by developing active immunotherapies or vaccines that can potentially treat or slow progression of MCI, mild cognitive impairment, or early Alzheimer's disease, or even go beyond that to prevent the disease to develop in individuals that are at high risk. What we are foreseeing that these vaccines would be like 2 or 3 injections intramuscular, with a like 30 to 60-day time intervals, and at doses that are orders of magnitudes less than monoclonal antibodies. So there will be more feasible and convenient for the patients. And to develop our vaccines, we are targeting the main hallmarks of disease, and in particular, tau aggregation and neuroinflammation. These two disease pathways seem to it is believed to work synergistically, one driving the other. And we have developed vaccines against these targets. We use a safe and proven virus-like particle platform that the technology has been developed at the University of New Mexico. So we design immunogenic peptides in silico and cross-link it to the surface of VLPs and use the final composition to immunize patients with the aim of activating B cells and producing disease-specific antibodies. The technology is protected by 3 issued and pending patents, and there are similar VLP-based vaccines already in the market. We have excellent safety, immunogenicity, and efficacy data in mice and non-human primates. For our lead vaccine, TB301, it reduces tau tangles in the brain, as you can see on the top left, that shows images of brain sections. The TB301 immunized animals had significantly less amount of tau tangles in the brain compared to control, and that corresponds very well with improvement in the memory in this mouse model of tauopathy or Alzheimer's. If you look at this graph, the immunized animals showed improvement in memory and increased rate of finding the target compared to the VLP control. We also have tested this vaccine in macaque monkeys, and showed that the peptides, the composition is immunogenic, and the antibody titer stays on for up to a year. And as we get similar result in mice as well. And also when we take serum from these monkeys and then we probe it against brain sections from human Alzheimer's patient, we see that the antibodies in the serum can find the target, suggesting target engagement. Our second lead vaccine that targets chronic inflammation is TB202. Chronic inflammation is known to be a major contributor to neurodegenerative conditions such as Alzheimer's or Parkinson, as well as neuropsychiatric disorders such as depression and schizophrenia. At the center of chronic inflammation is excessive activation of inflammasome. The diagram showing the inflammasome complex and production of inflammatory cytokines IL-1 beta and IL-18. So we have shown that when we immunize mice with TB202, we get reduction in IL-1 beta and IL-18, as well as systemic reduction of systemic inflammation. We have also evidence that some biomarkers of neurodegenerative diseases, tauopathy, are reduced in these animals, further linking neuroinflammation with neurodegenerative condition. So we believe that our vaccines are superior to competition, based so far, obviously based on the animal data that we have. We believe that our vaccines potentially more immunogenic because of the multivalency of the peptides displayed on the surface of VLPs. They provide higher specificity because we use disease-specific targets. They have also shown excellent safety, and our vaccines also provide potential to target patients at earlier stage compared to the other vaccines in clinical development. We are building a strong pipeline of single and combination immunotherapies for CNS and non-CNS diseases, with TB301 being our first asset, which is at IND-enabling stage. We are raising $7 million funding to complete our GLP toxicology studies for our lead asset. We have also opened a $1.5 million safe round as part of the $7 million round for early participants looking into get involved and help us to get closer to our major milestones. The main use of this safe round will be to optimize our formulation and dosing in non-human primates. We believe that we are well positioned to take advantage of recent comeback by Big Pharma to the space, and the recent surge in pharma deals around tau and inflammasome also validates our chosen targets and strategy. TheraVac is founded by leading scientists at the University of New Mexico, and on the business side, it's Laurent Valency and me. And we have years of experience in biopharmaceutical industry, taking drugs from discovery to clinical phase. Our clinical advisors, lead clinical advisor, Dr. Paul Scholz, is a director of Neurocognitive Disorder Center at the University of Texas Health Science Center. And lead business advisor is Mark Lebanon, former president of Sanofi. So this is the list of our partners and collaborators. Thank you for your attention. I'll be happy to take any question. David Carlson, our Director of Business Development, is attending in person. Feel free to approach him if you like to get more information on TheraVac. He's hard to miss. He's like a tall guy, probably the tallest guy in the room. Final note that we had the first key opinion leader events yesterday, and the information, the replay of this event is going to be available on our website, or you can reach out to either me or David for a copy of the video. Thank you, and I'll be happy to take any questions.
Questions for TheraVac?
I have a question.
Yeah.
Thanks, Ali, for the presentation. So the immunogenicity and efficacy studies that you showed in your previous slides. Just to clarify, was that for the lead asset, and is that non-GLP?
Yes, that's correct.
Okay.
Yeah, it's still non-GLP. Yeah.
Okay. And so the raise that you're looking for now is to do the GLP and
The raise we are doing is actually for further. Let me go to the slide. Yeah, it's actually no, it's still non-GLP. We are testing because we have different phosphopeptides. We want to make sure we want to take the best combination to the clinic. So we want to actually run additional tests on non-GLP in mice and NHP. Also determine the best dose that would be adequate for just long-lasting immune response. Before we do the next round, the next raise would be the additional 5.5 million. With that, we are going to be starting our GMP manufacturing and completing our GLP toxicology for TB301.
Okay, thank you.
Yeah, you're welcome.
Any other questions? Okay.